l'objet d'un contrôle de qualité le plus strict qui soit, du dossier de l'autorisation marketing aux multiples contrôles évoqués, la procédure mise en place. C'est par ça que nous allons ouvrir le symposium.
Alors, c'est mon plaisir d'introduire en anglais maintenant Dr. Koen Brusselmans. He obtained his PhD in 2001 from KULeuven in the Laboratory of Pharmacy Technology and Gene Therapy with Professor Desiree Bolen and Peter Family. Before that, he also obtained a master in cell and gene biotechnology from KULeuven. From 2000 to 2008, he was a postdoctoral researcher in KULeuven in the experimental medicine and endocrinology laboratory with KULeuven and Johan Sweden. He worked on lipid metabolism in cancer, and from 2008 and now he is an scientist expert and quality assessor for biological medicine at Sciensano. And he is also an alternate member for Belgium in the Biologics Working Party at the European Medicine Agency in Amsterdam. So, Dr. Brusselmans, you have the floor.
Good morning, everyone. Today I'm going to tell you a little bit more about the quality control of vaccines and the specific roles of manufacturers, regulatory authorities, and official control laboratories. First, a short disclaimer. This slide tells you who I am, what I do, and I'm going to tell you it's my personal view and may not always necessarily reflect the official position of the European Authorization Vaccines Agency, although I think for most points there will be not that much divergence. So as you may know, after having access to clean water, vaccination is actually one of the most important factor for saving lives worldwide and preventing infectious diseases. And therefore vaccines are among the most widely used medicinal products. And to give you an example, in Belgium we are using more than 1 million vaccine doses each year. Since vaccines are meant to prevent diseases and not for treatment, they are mainly administered to healthy individuals and mainly to children, newborns, and elderly, who are the more vulnerable people in our society. So, needless to say that the quality of vaccines is of utmost importance. We need to guarantee that they are safe and efficacious. And that's why we have quality control of vaccines at several levels. And I think we can say that in Europe, vaccines have the most extensive and stringent quality control system of all medicinal products. And I'm going to try to illustrate this during my presentation. In Europe, we have actually 4 levels of quality control. The first one is the evaluation of the registration file before the approval by the authorities. Second one is the quality control testing by the manufacturer on each single batch of vaccine. Third one are the GMP inspections, which are performed before the approval and after the approval on a regular basis. And fourth level is the additional QC testing by independent laboratories. Now we'll go into detail each of these. First, the evaluation of the marketing authorization file. So before a medicinal product can be used, the registration file has to be submitted by the applicant to the competent authorities. In the past, it used to be national agencies. Nowadays, this is mainly dealt with at European level by the European Medicines Agency. And the authorities will then perform an in-depth evaluation of all the information included in the file. And during this procedure, the assessors can ask questions, ask additional information, which the company has to provide, and this is then also included in the file. Only when all the data are complete and found acceptable, the registration file is finally approved. And this registration file actually contains 3 main parts, which are included in module 3, 4, and 5. The first one is the quality part. This contains information on the manufacturing process, on the product and quality control. The second part is the preclinical part that contains information on pharmacodynamics, the action of the drug, pharmacokinetics, how the drug is being absorbed, distributed, and metabolized in the body, on toxicology, and this is typically analyzed using in vitro tests and animal tests. And then the third part is the clinical one, also very important, of course, that contains the data from clinical studies in humans to demonstrate the efficacy and the safety of the vaccine. I will focus only on the first one, the quality part. So what is included in this module 3, quality part? As I said, information on the process, info on the manufacturer, all the production sites that have been used, a complete and detailed description of the manufacturing process, including all the process controls. Then we also need data in process validation. The company has to demonstrate that the process is capable to operate within a fixed set of process parameters. to make sure that it's capable to produce a product of consistent quality. Company has to demonstrate and validate that any residual impurities are removed to sufficiently low levels to make sure the vaccine is safe, of course. They need to provide information on the quality control of all the materials that they are using for the production. So they have to test for content, purity, microbial contamination, and if they are using materials of biological origin, they also have to check possible virus contamination. The file also contains information on vaccine composition, physical-chemical characteristics, and any residual impurities. As such, which need to be sufficiently low to make sure the vaccine is safe. There's also information on the container, the material of the container, the vials, or the syringes, to make sure there is no leaching of elements from the material into the vaccine to guarantee it's safe. Then, very important, the specifications. This is actually a set of quality parameters like purity, activity, pH, integrity, which are being analyzed using a fixed panel of validated test methods. And for all these quality parameters, the company has to propose acceptance criteria, so that means lower and upper limits, that need to be clinically qualified. That means if you are measuring the activity of the vaccine, and if it's close to the lower or the upper limit, it should still be guaranteed that those levels are still safe and efficacious. We also need a detailed description of all the methods used to test the specifications and the method validation to make sure the method is accurate and reliable. In practice, that means if a company is testing a vaccine sample today and another operator is testing the same vaccine sample tomorrow, we expect, of course, more or less the same result. Otherwise, the method is not reliable and not acceptable. Then we also need the actual test results from several batches of the active substance or drug substance and the final drug product at the time of release after production, but also at different time points during the shelf life. Those we call the stability data. For instance, if you have a shelf life of 3 years for a vaccine, you have to make sure that at the end of the 3 years, when you perform the testing again, that all these specifications still fall within the clinically qualified acceptance criteria. And so the registration file is only approved if all the data are found sufficient and acceptable. And what is very important, companies, once it's approved, companies cannot deviate from the contents of the registration file. So that means they have to perform the process the way it's described, and they have to perform all the testing the way it's described. If you want to change something, you first have to ask permission, and then the authorities should evaluate, and only after approval they can implement the change. So that's the first very important level of quality control. The second one is the QC testing by the company. So as I said, each single batch, both the active substance and the final product, has to be tested by the manufacturer. What is an active substance batch? This is actually the yield of a single production run in a fermenter, which is then further purified and filled in containers which can be stored for several months or years. A final vaccine batch is either complete or partial active substance batch, which is being formulated with excipients, possibly also with adjuvants, and then filled in vials and syringes ready to be used. So all these batches have to be tested using a fixed panel of validated methods, as described in the file, and the results need to comply with strict acceptance criteria that have been clinically qualified. And this is a guarantee that all commercial batches that will be produced later can also be considered as representative of those batches used in the clinical trials and can be considered safe and efficacious. What testing is now performed by the companies? Typically first some general parameters like pH and appearance. Also the identity of the antigen is being verified, the activity of the antigen content or the potency, the purity or integrity of the antigens, and then some general safety testing like stability and endotoxin testing. And the methods that are being used, those depend a little bit on the specific type of antigen or vaccine. I'll give you a few examples. For the activity or potency of a vaccine, what companies used to use in the past were mainly in vivo assays and typically the challenge assays. And this is nowadays still being used for tetanus vaccine and diphtheria vaccine. So what they do is actually they are going to inject either vaccine samples in mice or positive control, which is a standard reference vaccine with no efficacy, or negative control, placebo. And then these animals will be challenged with the administered the toxin or the tetanus toxin or diphtheria toxin. The mice who received placebo, those who get paralyzed and die. The ones who receive the vaccine will be protected. And then the protection induced by the vaccine is compared by the degree of protection induced by the reference vaccine. And that way you can analyze the potency. And that used to be a very efficient way of testing potency of vaccine in the past, but there are also some disadvantages on the in vivo testing in animals. First, it's very costly, it's time-consuming. Typically, these assays have higher variability because you have the intrinsic variability of each animal in terms of immune response. And last but not least, it's not really animal-friendly. And that's why authorities recommend, and companies are following this, they try to limit the use of animals in the testing, and in vivo assays are being replaced by in vitro assays. And typically an in vivo assay is not replaced by one single in vitro assay, but by several in vitro assays. For instance, you can measure antigen content, but that should be complemented by measuring also the purity or integrity. And one example of such replacement tests are the immunoassays. Here you see an ELISA, which is immunolinked immunosorbent assay. This is typically used, these immunoassays, for recombinant and subunit vaccines, like vaccines for hepatitis A, hepatitis B, human papillomavirus, inactivated polio vaccines. So actually you have a plate that is covered with the capture antibody. You're going to add your vaccine sample. The antigen in the vaccine will bind to the first antibody. Then you're going to add a second antibody that contains an enzyme, which is capable to convert the chemical compound and thereby generate cellular reaction. So the more antigen you have, the more the second antibody that will bind and the more color that will be generated. That way you can quantify the amount of your antigen in the vaccine. And typically when the antigen is degraded, it will no longer bind and will not generate a signal, although in some cases, partially degraded antigen can still generate a result. And that's why we need additional assays to assess the integrity or purity. And this is typically done by, for instance, NZSH, gel electrophoresis, or methods like site exclusion chromatography. Another type of vaccines are the live viral vaccines, and we're talking vaccines against measles, mumps, rubella, varicella, rotavirus, and also the oral live polio vaccine. So the virus concentration in these vaccines we are going to measure using cell cultures. So different dilutions of the vaccine are being added to cells, and then we are going to observe the cell death, and the higher the dilution, where you still observe cells dying, the higher the concentration or the titer in the vaccine sample. And this is typically expressed as plaque-forming units per ml for tissue culture infectious dose 50%. Again, another type of vaccines are polysaccharide vaccines. Those are, for instance, the pneumococcal vaccines and Haemophilus influenza type B vaccines. Those contain polysaccharides as antigen, so of course you have to measure the polysaccharide content. Not only the content, but also the size, because the chain length of these polysaccharides is very important for the immune response. If the chain length is too short, then the immune response is suboptimal. Apart from the content and the chain length, also the conjugation to a carrier protein is important, and nowadays most polysaccharides proteins are being linked to a protein. And the advantage is that these combined antigens actually not only induce a B cell response, but also a T cell response. So the immune response is much better if polysaccharide vaccines are conjugated. Of course, you also have to verify then if the conjugation to the carrier protein is properly done. And then finally, degree of absorption on the aluminium. If aluminium hydroxide is used as adjuvant, typically the antigens are absorbed to the aluminium, which is essential for having an optimal immune response. Otherwise, again, it's suboptimal, and again, companies have to verify the degree of absorption of these antigens. And this is not only true for polysaccharide vaccines, can be true for other vaccines as well. So, regardless actually which analytical methods that are being used, the specifications of the vaccine will always contain a set of tests that allow to conclude on first the correct identity of the antigen content or activity, the antigen size, detected purity, and sterility and endotoxin. And depending on the specific manufacturing process, these specifications can also include additional tests. For instance, if you're using toxic compounds in manufacturing, for example, formaldehyde is being used to inactivate the flu virus in influenza vaccines, then of course you need to test at the end for any possible residual formaldehyde, which cannot be present, of course, in the vaccine. In case of inactivated vaccines, we have to make sure that the vaccines themselves do not contain any live virus or any live bacteria. So that's also typical of the testing specifications. That was the second level of quality control. The third one are the GMP inspections. GMP stands for Good Manufacturing Practices. And as I said, all production sites have to be inspected. First time before the actual approval of the vaccine, and after approval of the vaccine on a regular basis, typically every 2 or 3 years. And during these inspections, the inspectors will verify if the manufacturing process and QC testing performed by the company is in compliance with the registration file and with the legislation. So they will check compliance with GMP guidelines and the European Pharmacopoeia. They will check if there's full traceability of all the materials and equipment used during production. They will check if the quality of the raw materials is being properly tested as described in the registration file, if the methods have been properly validated, and if all the process controls are being performed, and of course if the results fall within the acceptance limits. This is typically information and the data that the inspectors are reviewing during inspections to make sure that the processes are operating properly and capable to produce vaccines of adequate quality. So I've talked about the first 3 levels of quality control, which is the review of the dossier, the QC testing by the company, and the inspections. And I'll now say a few words on the last level, which is the additional QC testing by the independent MCL laboratories. In the EU legislation, there is a possibility for member states to implement an additional level of control. And this is not for all vaccine products, it's only for vaccines and also for plasma-derived products. And in most EU member states, including Belgium, this additional level of testing is mandatory, which means that each batch of vaccine that has been tested by the manufacturer has to undergo a second testing and release, that we call OGABR, by an independent laboratory, which is called ONCL, before the batch can be released to the market. And OGABR stands for Official Control Authority Batch Release. ONCL stands for Official Medicines Country Laboratory. And these ONCLs are actually nominated by the national authorities, by the government. And the network of all European ONCL laboratories is coordinated by the European Directorate for Quality of Medicines, which is located in France, Strasbourg. Now, this complete network of all these laboratories, which are located in more than 20 countries, they all operate under strict quality systems. ONCLs are inspected by other ONCLs and by EDQM. They can only use validated methods for the vaccine testing, and these methods are standardized based on the European Pharmacopoeia. If the test results for a particular vaccine batch are compliant with the specifications, then the ONCL will grant a batch release certificate, which is actually valid in all EU member states. There is a mutual recognition, meaning that one vaccine batch only has to be tested in one country, for instance in Belgium, and then other countries like Germany and France will also accept this testing and this certificate, and the other way around. And the Belgian ONCL is located within Sciensano, and it's one of the most important ONCLs for vaccines in Europe. What testing are we actually performing in the ONCLs? This is actually decided and defined by the EDQM and UMC network. For each type of vaccine, they will define a particular set of tests which are mandatory. It's not a complete set of specifications that the companies are testing, but it's typically a subset of the most important parameters like identity, potency, purity. And so in practice, companies are sending their samples to the ONCL. Then the ONCL performs the independent testing, and in general, normally this occurs in parallel with the company, so at the same time, meaning that results are available at the same time, and that will actually speed up the release of vaccines to the market, because we often have supply issues and shortages for particular vaccines, so it's very important that vaccines are being released to the market as soon as possible. And importantly, ONCLs not only do the testing, but also verify the results obtained by the company. So that's an additional level of control. If all the results are fine, compliant with the specifications, then the ONCL will grant a batch release certificate. You see an example on top of this slide. If the results are not compliant, if for instance the activity is too low, purity is too low, then a non-compliance certificate is issued, and this particular vaccine batch has to be destroyed. So, to release a vaccine to the market, a particular batch, the test results from both the manufacturer and the ONCL must comply with the acceptance criteria of the specifications, otherwise a batch cannot be released. So we can conclude that the OGABR testing by the ONCLs actually is a second independent QC testing that provides additional guarantee for vaccine quality and as such also for vaccine safety and release. And then finally, to give you an idea, in our ONCL at Sciensano, how many batches do we test? We actually test vaccines from 5 different vaccine manufacturers, in total 30 different vaccines. As you see, about 1,000 batches each year. The number has been slightly decreasing, not because the overall doses, the number of doses of vaccine has increased, but because the batches themselves have become larger. They contain more vials or more syringes, because manufacturers have been optimizing and upscaling their processes. In the meantime, we're also releasing and testing COVID vaccines, and due to that, the number has increased again to 1,200 to 1,300 per year. So at the end, I would like to conclude again that we have 4 levels of quality control in Europe. The first one is the review and the approval of registration file, which guarantees a strict control of the process and of the materials that are being used. The second one is the quality control performed by manufacturers, where the results should fall within clinically qualified limits. Third one are the GMP inspections, where the inspectors verify the process is performed and QC testing is performed in accordance with the registration file. And the fourth one is the independent testing by the OCLs. And this combination of control measures actually makes that vaccines have the most stringent and most extensive quality control system of all medicinal products in Europe. This was my last slide. I hope this presentation gave you some more insights and better understanding of how we can guarantee quality of our vaccines in Europe. I thank you for your attention. If you have any questions,
I have not said it in the beginning, but we have a very tight schedule. So each conference will be 20 minutes. I should have said it at the outset, and we have 10 minutes for questions. Now we are a little bit out of time, so I'm looking for some very focused questions in the audience here first. Short, precise, crisp questions. And if we don't have questions in the audience, I will move to the digital world.
The digital world keeps having some troubles with the sound. Je suis désolée pour ceux qui sont sur Internet et qui me signalent les problèmes récurrents, qui me disent ne pas entendre les orateurs, entendre les échos, etc. Notre manager technique est en train d'essayer de résoudre le problème, c'est tout ce que je peux vous dire. Désolée. Est-ce qu'il y a des questions sur Internet? Vous m'avez encore rien signalé, donc si vous en avez, merci de me les envoyer. Toujours rien dans la salle. Alors voilà.
Oui, c'était plus un commentaire.
Alors on va vous donner un micro, peut-être que ça va aider, on ne sait jamais, hein, peut-être que ça va fonctionner.
C'était plus un commentaire parce que je dois avouer que je ne connaissais pas, à titre personnel, ce double contrôle systématique sur chaque lot par les autorités. Et je pense que le grand public ne le connaît encore moins. Et pour rassurer le grand public, je me demande s'il faudra qu'il y ait une communication pour expliquer ce qui est fait et qui renforce effectivement la confiance dans les vaccins quand on connaît aujourd'hui une certaine reluctance, excusez-moi, par rapport à la vaccination.
Non, c'est effectivement correct que la plupart des gens ne sont pas au courant de ce deuxième niveau de testing. Et je pense qu'on a peut-être raté une possibilité avec le test Covid pour mieux communiquer ça au grand public, surtout pour les vaccins Covid en fait. On est en train de regarder maintenant comment on peut communiquer ça d'une meilleure manière au grand public, mais je suis tout à fait d'accord que c'est très important parce qu'il y a beaucoup de gens qui pensent toujours aujourd'hui que la qualité des médicaments est uniquement vérifiée par les firmes pharmaceutiques, mais il y a effectivement une deuxième contrôle indépendant qui doit assurer que effectivement la qualité est garantie. Vous avez tout à fait raison que on manque de la communication au grand public.
À quelle époque l'independent testing by OMCL was done? When did it start?
Ça a commencé, donc le réseau des OMCL, ça date des années 90, je pense 94. L'implémentation dans la législation européenne, ça date de 2001, si je ne me trompe pas. Mais donc au moins 20 ans, on fait le testing des vaccins. Plus de 20 ans en fait, on fait déjà le testing de chaque autre vaccin.
Oui, on a une question dans la salle.
Je travaille chez GSM. Et donc je voulais dire quand même que nous, en tant qu'entreprise, on communique toujours, en plus des contrôles qu'on fait nous-mêmes, on communique aussi des autorités qui sont vaccins. Mais je pense que dans le contexte du Covid, les gens ont eu peur d'une technologie pour laquelle il n'y avait pas suffisamment de recul. Je ne pense pas que c'est la qualité des vaccins qui inquiète les gens, c'est plus le recul pour la technologie AFMPS. Moi j'avais deux petites question pour vous. Est-ce que les vaccins dont vous avez parlé ici, on parle uniquement des vaccins humains? Première question. Ou est-ce que les vaccins vétérinaires sont soumis à la même réglementation? Et alors, vous avez cité 5 producteurs de vaccins en Belgique. Je veux juste savoir lesquels c'était. Hormis, je l'espère évidemment.
La deuxième question, c'est confidentiel. Mais donc, il n'y a pas beaucoup de producteurs de vaccins dans le monde, donc je ne peux pas donner les noms. Première question, pour les vaccins vétérinaires, on a aussi un testing au KBR, mais ce n'est pas le même concept dans le sens on ne va pas tester chaque lot et pas chaque vaccin. C'est plutôt une approche du risque. On va sélectionner certains vaccins chaque année et on va tester certains lots pour les vaccins vétérinaires. Pour les vaccins humains, on va tester chaque lot. Ça, c'est la différence.
Pas d'autres questions de cet espace digital. Je crois qu'on peut éliminer ici.